Hi, Dr. Harper. I hope all is well. For my presentation, I am looking at TGF-beta, which TGF-beta is just a protein that is found in your body. And the reason why I called it the controller of all cells is because it's responsible for a bunch of different actions that cells perform, such as whether a cell grows or whether it remains dormant or whether it undergoes apoptosis. Just your basic cell functions. And so I'm going to go over just the general overview of TGF-beta and then how it works with a gene called BMPR2 to create these or to signal for these cell actions to happen. And I'm also going to discuss how mutations can lead to diseases that can be linked to TGF-beta. So I'll go ahead and get started. So for a general overview of TGF-beta, so TGF-beta is just a multifunctional cytokine protein. So pretty much that just is big word for the family of proteins that it is found in. And so, as I said before, it's responsible for multiple cell processes such as cell proliferation, so whether or not the cell grows or stops growing. Cell dormancy, so sometimes cells just stop growing and they remain dormant, and so that would just be cell dormancy. And then also cell apoptosis, which is just a process where cells decide to pretty much self-destruct and stop growing, usually due to an overgrowth. That's when apoptosis usually happens. And so there are three different forms of TGF-beta. TGF-beta 1, which can be seen here, TGF-beta 2, which can be seen here, and TGF-beta 3, which is here down below. And so all of these different forms pretty much work together to signal the phosphorylation of this SMAD2/4. And so all of these have different structures and they have different ways of causing this SMAD2/4 phosphorylation, which eventually leads to gene expression. However, they all work together to do that. And so as you can see, they all have different structures, which actually the different structures is what causes their different functions and different roles in causing this phosphorylation. So, sorry, I forgot my note card. Let me just tell you the different functions of the TGF-betas. So TGF-beta 1 is the most abundant isoform of TGF-beta, and it is responsible for the growth of different cartilage, endochondral, and membrane, bone, and skin tissues. So it's responsible for regulating those. And then the TGF-beta 2 is otherwise known as the glioblastoma-derived T-cell suppressor factor, and that is responsible for tumor growth, and it can be found in neurons. And then TGF-beta 3, which I said can be found here, this is responsible for palate and lung morphogenesis. TGF-beta 3 is actually 80% amino acids from TGF-beta 1 and TGF-beta 2, which I thought was pretty interesting that it's 80% matches of a combination of both of those two. So that's just a general overview of what the different TGF-betas are and what TGF-beta does. So this is a normal cell pathway of TGF-beta. And so like I said before, TGF-beta actually works with a gene called PMBR2, which can be seen here. And so what TGF-beta does is it signals for the phosphorylation of this SMAD2/3. And so when this SMAD2/3 becomes phosphorylated, it attaches to SMAD4, and then this SMAD4, this combination is what leads to gene expression. But TGF-beta is not the only thing that causes this gene expression. BMPR2 works with TGF-beta, and they have like a relationship where they kind of coexist, and one of them regulates the other one, in leading to this gene expression. So BMPR2 is responsible for phosphorylating the SMAD158, and when that's phosphorylated, it also combines with the SMAD4 and this combination also leads to gene expression. So in normal cells, both of these two work together to regulate gene expression. And usually that gene expression is the one that leads to, like I said, cell proliferation, cell apoptosis, or cell dormancy. So, the general kinetics of TGF-beta. So this structure just basically tells you the general kinetics of the different spots of TGF-beta, including its phosphorylation, and then the phosphorylation of the SMAD2/3, and then its combination with SMAD4, and then obviously that leads to the different outcomes, and it gives you your half-life and also your Ks. And so I read an article that pretty much analyzed this because this is a bunch of jumble. And so pretty much from the kinetics of TGF-beta, it's actually really difficult to figure out the kinetics of TGF-beta, just because it is hard to figure out the cellular conditions of TGF-beta in humans. And so there hasn't been a whole lot of studying on the kinetics, but from what we do know is that these are the parameters that have been established. And then interestingly enough, research in these kinetic parameters actually found that changing the concentration of this TGF-beta, the overall concentration, does not change the kinetics of the overall pathway. The thing that causes a change in that kinetic pathway is actually a change in the molecules of TGF-beta per cell. So instead of the overall concentration, it's the molecules per cell, which is super interesting. So an increase in the molecules per cell, not the overall concentration, if you increase that, then your pathway is going to increase. Your rate is going to increase because your body is going to need to use that TGF-beta faster and faster and faster because there's such an overabundance of it in your cells, which causes this pathway to increase. And the rate of it to increase as well. And so the other thing with that is super interesting is that the rate of this pathway is actually time dependent. And so scientists did a study on this to see what would happen if you changed the molecules per cell, molecules of TGF-beta per cell for a short period of time compared to a long period of time. So they found that in the short period of time experiments that the kinetics, the rate of this TGF-beta signaling pathway increased slightly, not a whole lot. It wasn't super significant. However, when they introduced an increase in the molecules of TGF-beta in cells by a great number for a long period of time, the kinetics of this increased greatly. Which showed that it is time dependent, and in order for the reaction to speed up quickly and to be the fastest, it has to be exposed to a large amount of TGF-beta molecules per cell for a long period of time. So I just found that super interesting to look at that aspect of kinetics and understand kinetics in that way. So there are some problems associated with TGF-beta. The first big one, which is what I looked at for my research, is pulmonary hypertension. And so there are two steps to pulmonary hypertension that genetically can be linked to pulmonary hypertension. So the first step, we have a mutation in the BMPR2 gene, which like I said, works with TGF-beta. And so the mutation leads to an underexpression of the TGF-beta or the BMPR2 gene, which ultimately leads to a increase in the signaling of TGF-beta. And so I have a figure for this. So pretty much what happens is a mutation here causes a decrease in this phosphorylation pathway. And so you no longer have this here. There's not this here anymore. So to make up for this, since these two work together, you have an increase in the phosphorylation here, which leads to an increase here, which leads to an increase in this gene expression, which ultimately causes cell over proliferation. That's a mouthful. And so pretty much what happens in pulmonary hypertension is you have smooth muscle cells and endothelial cells that build up in tissues and in your arteries, and that's what causes the pulmonary hypertension. Well, this can be found to be linked to an increase in this TGF-beta due to a mutation in this BMPR2. So this has caused a whole lot of problems in many different people because this TGF-beta is being overexpressed and is working overly hard to make up for this lack, which causes pulmonary hypertension. Another problem associated with TGF-beta is with cancer, and so a lot of cancers can be caused from the same pathway. And so, like I said, again, you have a blocking in that BMPR2, which ultimately leads to a increase in that phosphorylation of the SMAD from the TGF-beta over here. So normally in normal TGF-beta, SMAD phosphorylation, you have a tumor suppressor. And so when there's normal phosphorylation, there's no tumors present because the TGF-beta is ultimately suppressing that tumor. But when you have a cancer cell and you have cancer and that BMPR2 is mutated and is turned off, this leads to problems because your TGF-beta overphosphorylates again, instead of being a tumor suppressor, it leads to a tumor promoter. And so that can lead to cancers such as liver cancer, lung cancer, breast cancer, all sorts of different cancers in the human body. And so the pathway is very similar to that of pulmonary hypertension. And so as you can see, different diseases can be caused by problems in this TGF-beta signaling pathway. So pretty much in conclusion, as I said before, TGF-beta is very, very important in multiple different processes in the body. It is the thing that acts on cells that tells them to, hey, you need to either proliferate, or you need to stay dormant and stay how you are, or you need to have apoptosis occur. However, a lot of studies have been done and found science that links to problems such as cancer and pulmonary hypertension with a general increase in the signaling of the TGF-beta pathway. And so those SMADs that TGF-beta normally phosphorylates at a normal level gets increased due to a lack in the BMPR2 SMADs, which ultimately causes a buildup of cells, which can either lead to pulmonary hypertension or cancer. There could be other diseases. I didn't find any other diseases when I was doing my research, but that could be a further area of research. Also, like I said, there is not a whole lot of research behind the kinetics of TGF-beta, and so I'd be super interested to see if people have done more studies, or super interested to see if people would do more studies to establish the kinetics of that TGF-beta, because like I said, the conditions, the parameters that are there have been established, but they're not super well known, and so it's not like super big in research. So I'd be super interested. That's an area of research that needs to be more explored and more looked into. And so this figure right here is just an overview of everything that TGF-beta and also BMPR2 controls. I just went over the SMAD pathway, which leads to gene expression, but as you can see, TGF-beta and BMPR2 working together is super important in a bunch of different phosphorylations and a bunch of different pathways. And so that actually is an area of research that I'd be super interested to look more into. And so so that's pretty much a general overview of what I wrote my paper about and of the TGF-beta pathway. So here are my references. I have 2 slides of references. I use a lot of references. And then, so thank you, Dr. Harper, for listening to my presentation. If you have any more questions, feel free to reach out to me, email me, contact me about that. I did do research looking at the drugs to target the TGF-beta pathway with pulmonary hypertension. So if that interests you at all, feel free to contact me about any questions. I didn't get to do the research because of COVID, but I did set up an experiment and start preliminary research. So if that interested you at all, please don't hesitate to contact me. I hope you have a wonderful Christmas break, and I thank you for your time.